the structure and mechanism of the mammalian fructose transporter GLUT5. So through question 1, we want to introduce the GLUT transporters, their general function, the isoforms of GLUT1, GLUT2, GLUT4, GLUT5, and the tissues in which they are most highly expressed. We will then describe in the second portion of the question, describe the kinetic properties and specificity of GLUT1, GLUT5, GLUT2, and GLUT7 for fructose, glucose, and galactose. So what is GLUT? It is abbreviated as glucose transporters, in which they come from a family of solute carrier 2 proteins, and they allow for glucose, fructose, and galactose to enter the cell down a gradient. And as you can see in the upper right corner of this slide, you'll see an image where you'll see a transporter called a passive carrier protein showing glucose solutes entering the cell down a gradient. This is a simplified version of it, but there's several mechanisms, several conformations in which the glucose transporter will allow the solute to enter the cell. Passive carriers do not require any energy expenditure by the cell, but it is slower than that compared of channels because passive carriers have to go through several conformational changes. These conformational changes will be further elaborated later in the slides. Glucose transporters, or GLUT, has 14 different isoforms. So for example, GLUT1, GLUT2, up to GLUT14, all of which are uniporter except for GLUT13. So there are different isoforms that are mentioned here. I will say specifically where they mostly reside in. There's GLUT1, which mostly resides in the brain-blood barrier, as well as the red blood cells. GLUT2 transporter mostly resides in the liver, kidney, small intestine, as well as the pancreas. GLUT4 transporters, stimulated by insulin, are mostly resided in the skeletal muscle, cardiac muscle, as well as the adipose tissue. Further emphasized later in the slides, we have GLUT5 transporter, which transports fructose and mostly resides in the small intestine. That's pretty much it for me in regards to my presentation. I know it's a little short. I'm around 2 minutes and 50 seconds, 53 seconds, so I'm okay with it. My classmates will handle this slide and that slide and this slide. Yeah. We can go over some cards real quick just so I can, I don't feel like I'm doing nothing, you know. I want to feel like I'm doing something today. I will be going through all of these cards ideally by the end of today. I just need to work. I just wanted to make sure my presentation was kind of decent enough, right? Presentations take some time because you want to be able to articulate well, articulate loudly, and to provide information and do proper research on whatever you're talking about so that you can answer appropriate questions or maybe inappropriate questions. I don't know. The fifth amino acid, the ALA, part of the pentapeptide chain. Oh, this is for immunology. Well, this is actually microbiology. NAM is N-acetylmethamine or something like that. That was initially created from a cell wall biosynthesis, is a target for antibiotics. So this is a very detailed mechanism in which the fifth amino acid right here will be deleted so that antibiotics won't be able to attack it anymore. That's what I think. But yeah, the fifth part is a target for antibiotics. But if we remove that, the antibiotics can't have access to it, which is, I guess, is a bad thing. But the cell, you know what? I need to do more research on this because I don't know where this really pertains to. Like, I don't know if it's in the human or... Oh, it's in the bacteria, right? Bacteria. So it's going to have some form of resistance, right? I guess that's how it becomes resistant to antibiotics. Um, Honestly, the questions I made, these ANKI cards are really, I think they're done well. I pat myself on the back because some of these questions, I mean, these ANKI cards I think they're done pretty well compared to what I've done in the beginning of the semester. This is because I'm getting used to the classes and I know my pacing. So epinephrine comes from tyrosine, right? If you recall correctly, we start off with phenylalanine, making tyrosine, making L-dopa, making dopamine, making norepinephrine, and then epinephrine. Okay, and so tyrosine can eventually make epinephrine, and tyrosine is stored in secretory vesicles called, I think secretory is spelled incorrectly by the way, but they're stored in vesicles called the chromaffin granules. Something to know. Protons will be accumulated in the intermembrane mitochondrial space. So if you remember in the electron transport chain, you'll remember that there are several complexes that will pump proton into the intermembrane mitochondrial space, but then the ATP synthase, the complex 5, will pump proton into the inner mitochondrial matrix. But the accumulation happens in the intermembrane mitochondrial space, which is very important. And this is true. These are just semantics. It seems that AMP will be increased as a result of this being increased. I'm assuming that this is probably hexokinase or glucokinase. So we have muscle, right? Muscle. I'm going to say it's hexokinase because I think glucokinase comes from the liver, while hexokinase comes from many tissues, including muscles, right? The reason why I said this is because I thought this was glucose, which converts into glucose. You know, I've been mixing things up. In the back of my peripheral vision, I kept thinking that maybe this is glucose, going through the glycolysis pathway because I saw GLY, well GL and then pyruvate. So this does make sense in some ways. I don't actually remember this yet, so I'm going to put this as a 1. I think that's it for today. I'm going to stop and go to school and do the presentation, come back home, and then do some ANKI cards with you guys. If you guys are willing to go through it with me and see how I use these cards, see how I learn from these cards, because making cards, yeah, sure you learn it, but do you retain it? And that's where ANKI will use its algorithms to then help you. That is all.